pleasure of introducing Dr. Deanna Kulpa. Dr. Kulpa joined Emory in 2016 and is an associate professor in the Department of Pathology and Laboratory Medicine in the Emory School of Medicine. And she received her PhD in human genetics from the University of Michigan in 2005, and then subsequently completed her postdoctoral fellowship there in 2012. She is a faculty mentor in both the MMG and IMP programs, and her research is focused on defining the mechanisms that promote HIV persistence in anti-retroviral therapy or ART-treated HIV-infected individuals. Her laboratory employs novel in vitro, ex vivo, and omics approaches to identify pathways and mechanisms that better our understanding of HIV latency, establishment, and maintenance. The goal of the Kulpa Lab is to identify and evaluate therapeutic strategies to counteract these mechanisms, which will constitute the basis for the design of clinical studies aimed to eradicate HIV. Welcome, Deanna, and to the IDAS seminar series.
Thank you very much, Rachel. So I need to share my screen.
Yes.
Is that good?
Would you like to move the black bar up there?
Oh, yeah.
And yeah, high floating media control. High floating media.
We'll show them.
Yes.
Okay. All right, so good morning, afternoon, sorry, trying to figure out what time we are still. So today I'm going to talk a bit about some of the work that's going on in our lab and introduce you as well to what. There we go. Introduce you to myself. I was requested to give a little bit of background information on me. So I just wanted to say first, I was born and raised on a farm in northern Michigan. So basically where I grew up, not only was getting an advanced degree something people didn't do, they also didn't go to college. So and when I was in high school, I didn't actually plan on going to college. I didn't really know what I was going to do. And it was one of my teachers in my senior year of high school who asked me, what are you going to do with your life? And I said, I think I want to be a writer. And she said, well, how are you going to do that? I said, I don't know. And so she said, you need to go to college. And I said, okay. She said, you need discipline. That's what you need. I was like, all right. So I took her advice, and at the time I wanted to be a writer. So I looked in the state of Michigan for schools that had a English degree with a writing emphasis. And there was one, Michigan State University. So I applied to it and I went there. And again, this was basically, I don't know what I'm doing. I'm walking around trying to figure out what college is and even, you know, what I wanted to do while I was there. And somehow I ended up on the archery team. And one of our coaches was the dean of the College of Natural Sciences. And, you know, I really enjoyed science. I always loved science, but there were conversations, you know, one of the things that occurred to me was the way science was presented in school was, this is what we know. We know this, we know this, we know this. And it was never a, we don't know stuff. You know, and so science never occurred to me as a career because it was always like, well, we already know everything. And therefore, what is there to do? And so after having conversations with him, where it's more like we're just talking about our love of science and how actually there is a lot to do. There's a lot we still need to know. He actually convinced me to change my major, and I did. And so I ended up getting a bachelor's of science from Michigan State in microbiology immunology. And then, but after that, again, it was, well, now what do I do? Again, being sort of first generation, sometimes you really don't know what you're supposed to do now. And so what I did was I got a job as a lab technician, spent a few years at the University of Michigan in Alice Telzinski's lab. And from there, I decided I wanted to go into biotech. And so I moved out to San Diego and worked for the Chiron Center of Gene Therapy for 3 or 4 years. And then kind of hit a wall. You know, in terms of what I was interested in terms of science and what I wanted to do, there was sort of the, well, now you can be a manager, you know, and really it was, again, someone that I worked with, Mark Mitchum, was a very talented biochemist who did not have a PhD. And when we talked about where am I going, what am I doing? He said, Deanna, what you want to do is lead research. That's what you're interested in. And in order to do that, you need a PhD. It's like, I can tell you, I don't have a PhD, but I'm smart. And everybody I know, works with me knows I'm smart. But as soon as I have to walk into a new room, I have to prove myself all over again. It's like having those three letters gets you just like farther ahead, like go to graduate school. So again, I'm just doing what people tell me to do. So I applied to graduate school and somehow managed to end up back in Michigan again. I got my PhD in human genetics, studying non-anti-retrotransposons in the human genome. That I did a postdoc with Kathy Collins, also at the University of Michigan, where I really started to get into HIV research, looking at the mechanisms of HIV immune system evasion. From there, I went down to Florida to work at the Vaccine and Gene Therapy Institute of Florida with Rafik Sekali, who is now here. He followed me. I actually came. And it was there I really started to get into HIV cure. And working with Rafik has been just a real highlight of my career in the fact that he really showed me what collaborative and integrative research programs look like. A lot of the labs I'd worked in had been like, here's our project, we have a little quarter, we do a little thing. But Rafik, if is just like he's a monster, right? I mean, everything, he's connected to industry, he's connected to nonprofits, he's connected to academia, he's connected to all of it. And so it really kind of showed me that in order to really tackle a problem like HIV, working in this big team environment was really the way to go. So ended up being able to establish my own lab here at Emory in 2016. And then just a couple of years ago in 2020, we linked up with Mirko Piardini and Guido Silvestri to form one of the Martin Delaney collaboratories for HIV research, and ours is called Erase HIV. And so this is a lot of the information I'm going to show you today comes from this collaborator. So broadly, what we do, we're focused on curing HIV through innovative immunological approaches, which everyone wants to be innovative, but we try to actually be innovative. We use in vitro, ex vivo, and in vivo models to study HIV pathogenesis and immunology. And one of the things, or several of the things that we're very interested in terms of like our approach to HIV cure is we're interested in studying the mechanisms of persistence, and I'll go through a bit of what I mean by that in a minute. As well as what we call the HIV reservoir, so understanding how we can quantify the frequency of HIV infected cells in a person. And then the immunology, we're very interested in the link between inflammation and exhaustion and HIV persistence, how mechanisms of immunological control of HIV replication kind of play into persistence. And we're also recently moved into a lot of T cell metabolism, which seems to be playing a big role in both the cells that are becoming HIV infected, as well as the immune cells that are trying to combat that infection. And then ultimately to move into therapeutic interventions that we can test. So just to kind of give everyone an orientation, anti-retroviral therapy just basically was a godsend to HIV infection. Initially, when it was first described and discovered, there was no treatment. And so a few of the drugs that got pulled out were basically off the shelf sort of things that were repurposed from other diseases. But as time went on, more and more compounds became available that were blocking specific events during the viral life cycle. So whether virus entry, reverse transcription, integration. So HIV is a lentivirus. So it actually makes a DNA copy of its RNA genome that becomes permanently integrated into the host cell genome. So once the virus comes into the cell, it doesn't just replicate out in the cytoplasm, it becomes a permanent resident of that cell. So then, and try to deal with it on the other side of it, we can try to block its transcription, translation of the proteins that it needs to make more virus, also as well as like inhibiting some of the proteases that are needed to kind of cleave and assemble the particle and capsid, as well as some budding inhibitors, so try to like keep the virus from actually leaving the cell. And so we have a whole number of drugs in our arsenal now that can target all of these. And the reason why we needed so many is because if you just try to target one aspect of the viral life cycle, we found is HIV, and when it replicates, it's very error prone. So when we look at making the DNA copy of its RNA genome, there's no proofreading that's associated with reverse transcriptase. So if it makes some errors, it just kind of goes, oh, okay. And it just kind of rolls with it. And what ends up happening is when you're making millions and millions of virions, you're going to end up with some that are going to be resistant to whatever you're using. So we found is if you use a combination of different compounds that can tackle different aspects of the life cycle, getting around all of them or 3 of them basically is what we use becomes very difficult for the virus to do. And one of the things that I wanted to make sure to do is to give a shout out to researchers here at Emory. So Raymond Shinazi and Dennis Leota were actually one of the ones that developed 3TC, which was one of the very first compounds that was used against HIV infection. And in fact, like I can't remember what the statistic is, but it's some staggering thing where if you look at the number of people in the world who are HIV positive who take anti-retroviral compounds, like 90% of them take a drug that was developed by Raymond Shinazi. It's just amazing what he's been able to contribute to the world of HIV research. He also went on to develop FTC or evitricity. So definitely some amazing work on HIV that's going on here at Emory. And we always like to acknowledge our great colleagues. So why was antiretroviral therapy? Besides the fact that we now know that it can really impact dramatically the lives of people who are HIV infected. We know that HIV infected individuals, when untreated, can go on to develop acquired immune deficiency syndrome. So HIV infects CD4 T cells, which are part of your immune response, and eventually through a number of different processes, we see the depletion of CD4 T cells, and you end up having a decline in immune responses. You lose your T cell health, and you end up developing AIDS. So one of the things that people thought when we were going to develop antiretroviral therapy was that all you really need to do is to take care of these cells that are producing the virus. And all you needed to do was to stop them from infecting other cells. So we know that productive infection of CD4 T cells with HIV results a lot in cell death. So eventually the thought was if you just put the individuals on antiretroviral therapy, which is the gray square, eventually these cells that are producing virus are going to slowly die out. And which if you can stay on antiretroviral therapy for long enough, eventually, all the infected cells will die out and you'll be the individual cured. What we now understand is that the virus can establish a latent infection. So right at the same time that at the very beginning of HIV infection, so I should just warn you, the y-axis is plasma viral load. So this is indicating this top peak is the acute infection. So the very beginning of the infection, you see virus coming in, replicating all over the place. And then we see the immune response kicking in, sort of lowering the viral load. And then we have kind of basically a chronic phase of the infection. So all during this part right here, where we see these virally infected cells are producing tons of virus, giving us a lot of plasma viremia. At the same time, there are cells that are becoming latently infected. And we know this because we can actually study individuals who are viremic. They have a ton of virus in them, and you can actually find cells that are latently infected right in those pool of infected cells. And so what happens is that when we introduce antiretroviral therapy, yes, all these big viral producers are they eventually die out. We lose the spreading infection through antiretroviral therapy is inhibiting new infection. But the fact is we have these latently infected cells that are invisible to the immune system. They're not making viral proteins. And so the immune system doesn't know they're there. And so it can't take care of them. And then they just kind of hang out. And what's brilliant about the virus is that it manages to get into cells that are long-lived. So it gets into memory CD4 T cells. And your new memory is meant to last your entire life. So the idea is that you can have a cell that, maybe over time could eventually differentiate and die out. But we also see the persistence of memory basically through homeostatic proliferation. So some memory cells, when they receive signaling from their cognate antigen, they don't become activated. They basically just proliferate. Their whole job is to say, oh, we've seen this antigen before. We're going to want some more of us around. So we're just going to make a few more of us as backups. And so what you end up seeing is clonal expansion. of an individual CD4 T cells that can be infected, and you can actually trace them through integration sites and things like that. So basically, the virus is persisting over time because of the cells that it's getting into, and the antiretroviral therapy doesn't really have a good way of dealing with those cells. All it can do is deal with cells that are actively making virus. And so then what ends up happening is if you interrupt antiretroviral therapy within a few weeks normally, we see virus rebounding, we see plasma viremia established all over again. And again, we're going to have a mixture of productively infected cells and latently infected cells. And one of the things I want to emphasize is that during when the individual is on ART, there's nothing about antiretroviral therapy that keeps cells from producing virus. It just keeps the virus from being able to go and do other things. But a cell itself that's latently infected can produce viral RNA or viral proteins or even virions at any time. And what we normally see is that you can get something called a viral blip, where you might see if you're monitoring a person on antiretroviral therapy, where we have a reactivation of an infected cell that can start to produce some virus. It's very small because that virus can't spread and start infecting neighbors, but it's enough that we can actually detect that there is some activity. So we know that this reservoir is not just all dormant; it's very dynamic, and so it's something we're very interested in is trying to really characterize what we call this reservoir. And this is the reservoir is just basically the frequency of infected cells that can contribute to viral rebound after ART interruption. And so this is our principal barrier for HIV cure research, is what to do about this population. And there's been a number of assays, including ones we developed in our lab, to try to quantify the frequency of these cells. Again, when they're latently infected, they're not expressing viral proteins. So how do you see them? They're rare, so we can see like maybe 1 in a million CD4 T cells are infected. So it's not like you can just take a sample of blood and see them all over the place. And so studying them from a person is really challenging. And there's been a number of assays and ways that people have developed to try to characterize them, study them, and better understand how the virus is persistent. So why does the frequency of infected cells matter? Why do we need all these assays? So we know in the absence of ART, there's a clinical correlation with the viral set point during the chronic infection and time to appearance of AIDS. So after our primary immune response, we have what we call, after the establishment of the chronic infection, you can basically see a spread in terms of the level of virus that we see as sorts of plasma viremia in untreated individuals. And what we can see is that an individual who has a high viral set point, and this is characterized as being over 100,000 copies per mil, they can actually develop AIDS within a matter of months. Where you see individuals that have a lower viral set point, this can be years to decades of how long they progress to AIDS. And the viral set point is really going to be immune mediated. But there is a correlation with how many infected cells you can find in a person that actually can predict this as well. And so why we're interested in reservoir size is when you have this information, it can help you on the other end of it when you're studying HIV latency and cure, how to predict how your therapy is going to work. And so in HIV cure, we're looking at basically 2 different strategies. One is eradication and the other is remission. So eradication is basically introduce some sort of intervention that can disrupt HIV latency, allow the immune system to start to recognize these cells, and basically eliminate all the HIV infected cells. So having a larger reservoir is going to be more difficult to eradicate. So you want the smallest reservoir as possible. And for remission, this idea is if you can't get rid of all the virus, can you have an immune system that controls it enough that the viral replication is not a problem? And so we can see this after hemolytic treatment interruption, or ATI, you see it referred to as, and then we measure the time to viral rebound. So basically the length of time it takes to go after you've stopped ART for the units you to see a virus in the plasma. And this time to viral rebound actually correlates with the size of the reservoir. So again, the larger reservoir can shorten the duration of the rebound. So we're very interested in the frequency of these cells simply because it can tell us a lot of clinical things, or predict a lot of clinical things. in terms of what we're interested in for HIV cure. So my lab is very interested in what are the mechanisms that are driving HIV persistence and latency. So if we're interested in the size of the reservoir and how these cells are being maintained, one of the things we really want to know is how and what can we do about it. Good question. Sorry.
Oh, no, sorry.
Okay. So our focus is to define the roles of CD4 and CD8 T cells in HIV persistence during PART. And so, as I mentioned, CD4 T cells are the main targets of HIV infection in vivo. HIV can also infect macrophages, dendritic cells, some glial cells that can be found in the CNS. But the primary targets are CD4 T cells. And one of the main immune modulators of HIV, the control of HIV replication, are HIV specific CD8 T cells. So we have an antigen specific arm of the immune system in the CD8 T cells that can recognize productively infected cells that are producing lots of viral proteins, and then can actually do something about them in terms of targeting these cells specifically for elimination. So the antigen-specific CD8 T cell responses are associated with decline in plasma viremia. And this is what I was mentioning earlier. When you see the initial dip after the acute infection, you'll see that is where the CD8 T cells are starting to emerge, is when you see the dip in plasma viremia. And it's these activities of CD8 T cells are very well characterized. So we've seen that HIV as well as SIV CD8 T cell responses are detected as early as 7 days after infection. So they appear rapidly. We see the expansion of these antigen-specific CD8 T cells do correspond with the decline in plasma viremia. So we know that they're involved in the control of the amount of virus we can see. And then we also can see that virus escape mutations are emerging in response to those immunological pressures by the CD8 T cells. So we end up seeing accumulation of mutants and the plasma viremia that basically will show an invasion of the CD8 immune response. And we also have individuals that were called elite controllers, where basically they have been characterized to have very potent cytotoxic T lymphocyte cells with high polyfunctionality, proliferative capacity, and killing potential. And it's believed that these very potent cytotoxic T cells are responsible for their elite controller phenotype. And when we say elite controller, what that means is in the absence of anti-retroviral therapy, they're one of those people who have very low or undetectable plasma viral load. They're not cured, because if you can take some cells and look for the virus, you can find the proviral DNA integrated into the CD4 T cells that you're looking at. But yet their immune system is keeping down and rapidly recognizing any HIV infected cell that starts producing virus. So these are very rare individuals, but we've identified them now, and we can study them. And one of the things we've identified is very specifically they have extremely potent CD8 T cells. But now there are also numerous studies that have shown that CD8 T cells from people living with HIV can secrete soluble factors that negatively regulate HIV transcription in CD4 T cells. And our own recent studies have demonstrated that CD8 T cells from HIV-naive individuals can reduce HIV expression during ART-free in vitro infection of autologous activated CD4 T cells. So what that means is we can take the co-culture of HIV-infected CD4 T cells, these cells have been infected in vitro, co-culture them with activated CD8 T cells. These are all HIV-naive individuals, so there should not be any HIV-specific CTLs. There's no expanded HIV-specific CD8 clones. These are just total CD8 T cells. And what we can see is we see inhibition of HIV viral expression in these CD4 T cells in our in vitro model system. But the mechanisms responsible for this we're still figuring it out. But we do know that this non-cytolytic CD8 T cell mechanism can regulate HIV transcription, can be really important as the establishment and maintenance of the latent reservoir in CD4 T cells after ART has been initiated. And so we can think of this non-cytolytic and cytolytic functions of CD8 T cells as being synergistic when it comes to dealing with a viral infection. So you either have HIV-specific CD8 T cells that can destroy these viral-producing cells, but if you can't kill all of them, maybe you can shut some of them off and you can actually get rid of virus production. So it's a regulatory mechanism as part of the immune system that gives us a way of dealing with a viral infection that we can't completely get rid of. However, ironically, these two functions have divergent effects on the HIV reservoir that can be magnified after ART initiation. Essentially, ART takes the place of HIV-specific CD8 T cells, and the fact that you no longer have the spreading infection. But you do still have this pro-latency mechanism around that can be influencing the persistence of HIV-infected CD4 T cells. And we showed this beautifully in a paper that just came out like a month or two ago. This year, where we looked at using a non-human primate model of HIV infection. So this is rhesus macaques infected with SIV. And what we did in this experiment, and this experiment was designed by Guido Silvestri. So our control group is essentially just infecting rhesus macaques with SIV, waiting 2 weeks, and then beginning ART. These animals are maintained on ART for 50 weeks before treatment interruption, and then we follow them through after treatment interruption, looking for viral rebound. But in order to understand the role of CD8 T cells at the very beginning of HIV infection, so if this idea is the first 7 days, we know that we're seeing in humans HIV-specific CD8 T cells emerging. Well, what if we take those away? What happens to the reservoir? What happens to the affected cell population? And you can guess that just by taking them away, well, gosh, you don't have that CD8 response. You're going to get virus everywhere. You're getting a ton of infected cells. So we thought we would look at 2 different time points. So one is taking away those CD8 T cells just before we add the virus. And another is infecting the animals, letting them go for 2 weeks. So enough to have some kind of CD8 T cell response, and then getting rid of the CD8 T cells right at the time ART is starting. So what that does, it'll help us understand. So what happens if you lose all of those HIV-specific CD8 responses right when you're starting ART? So basically, when we say that ART is replacing CD8 T cells, what happens when you basically are getting rid of those? What do you see? And what you may expect is that, okay, these are our different groups. The black is the control group, so without CD8 depletion. The pink is getting rid of CD8 T cells before infection. The teal, I guess, is getting rid of the CD8 T cells right as we're starting ART. And what you may expect is that, in fact, you see higher levels of plasma viremia in these animals when you take out the CD8 T cells. It makes complete sense because you're losing all those HIV, SIV specific CD8 T cells right at a time when you really need them here. And this is just a calculation of the decay rate. And so, this is looking at different time points, but you can see that the cells coming from the viral load coming from the control animals is declining much more rapidly, or significantly more rapidly than the animals that were depleted. But what's interesting is when we actually then characterize the frequency of infected cells, this higher plasma viremia was not coming because we had a ton more infected cells in these animals. And in fact, the frequency of infected cells in these groups are indistinguishable from each other all the way out through basically a year apart. So this lower plasma viremia in the control group can't really be contributed to the CD8 T cells killing the infected cells. So what is happening? What we end up seeing is that there's a couple of different populations of CD4 T cells. As I mentioned, when during primary infection, we see there's cells that are producing a ton of virus, there's cells that are like latently infected. And what we're seeing when we're moving the CD8 T cells is that they're really impacting the short-lived virus-producing cells. So if you're looking at this is a mathematical modeling that's done by Miles Davenport, one of our collaborators. He's looking at the decline rate of the short-lived cells, and you can see that the short-lived cells that are producing all this virus, they're declining and decaying much more rapidly when the CD8 T cells are around. And then when we remove the CD8 T cells, you can see these cells are living longer and basically just producing more virus. So that's why we see this kinetics. Interestingly, now when we're looking at what we call the latent cells, so the cells that now are going to stick around for a long time, these are actually producing virus for a lot longer too. So these are the cells that are going to establish the latent reservoir. They're actually expressing some RNA, even when the CD8 T cells are gone, meaning we have some sort of mechanism that CD8 T cells could potentially be pushing on these cells to basically shut down HIV replication or HIV expression. And so how does this work? So this was one of the things about what's fun about working with Mirko and Guido is that we can go back and forth between in vivo and in vitro models to try to understand. Asking mechanistic questions in a non-human primate is very difficult But using our in vitro model system, we can actually ask someone those very specific questions. And so to do this, we used a latency, HIV latency model that I developed working with Rafiq. We call this model LARA, for latency reversal assay. And this assay just requires using some fuzzy codes from HIV naive individuals, so there's no in vivo infection here. We take the memory CD4 T cells from these individuals, and then we infect them in vitro. And we use a replication competent clone of HIV that was, this is actually not a lab strain, it was one that was isolated from an HIV infected person. And then we use a combination of different cytokines and conditions over a 2-week period that will allow these cells to basically mimic the sort of signaling that they'll receive in vivo to establish a latent reservoir. So a number of the cytokines that we know are involved in this is TGF-beta. These are cytokines that really can shut down a lot of cellular transcription. We know that IL-7 is a big component of is homeostatic proliferation maintenance of these cells. And we also use a feeder cell line called HAEs. This is a glioblastoma cell line, secretes a number of factors, IL-10, TNF alpha, and these cells, when they secrete into the media, we combine that with TGF-beta and IL-7, and this ends up giving us a really nice population of latently infected cells at the end of our assay. We can get somewhere between 10 and 30% of the cells infected and then maintained on anti-retroviral therapy for these 2 weeks so that we have basically no HIV expression at the end. We keep these cells on anti-retroviral therapy for the entire time. So we're not getting a spreading infection. We're not seeing accumulation of mutations. Essentially, when we infect the cells, they're immediately put on sequinavir, which is a protease inhibitor. And what we're seeing is the virus can integrate and then it can't go anywhere after that. And so we just have a single round of infection. And we did a lot of characterization of these cells that we published in the JBI paper a number of years ago. And one of the things you can do with this assay is you can ask questions about, what can you do with these latently infected cells? Can you apply different compounds to them and ask questions about latency reversal? But you can also look at the establishment of the HIV reservoir at the time of ARC initiation, which really models that primate study. So this is what we did. And so in this model system, we take CD8 T cells from the same individual. So on day 0, when we enrich the CD4s, at the same time, we're also enriching total CD8 T cells. And in parallel, we're activating the CD8 T cells while we're resting the CD4s. And so here's our CD4 monoculture, and this would be our CD4 CD8 co-culture. So after we infect the CD4 T cells with HIV, we let the cells rest for several hours, and then we put them into co-culture with their autologous activated CD8s. And then they're maintained on ARC for an additional 3 days. And what we do is we're asking, okay, so now after these cells have been sitting for a few days, we expect some level of HIV expression. Simply because we've looked at these monocultures a million times, we see that if you look here, this is the expression of CD4 and CD8 by flow cytometry. You can see these are HIV T cell populations. We look specifically at this population for HIV GAG. So you see CD4 on the x-axis and HIV GAG on the y-axis. HIV expresses a number of proteins that down-modulate CD4 T cell surface expression. So when we're looking at HIV expressing cells, we're looking for CD4 negative GAG positive cells. And so you can see here, one of the reasons why we see this kind of smear of CD4 T cell expression is because this is coming from our CD4 low population. So we see a number of, in this case, it was like 7%, I think that number is, of cells that are expressing HIV GAG just after 3 days after infection in the monocultures. Now, interestingly, if you have CD8 T cells from the same donor, now you co-culture together, and then you look at the frequency of infected cells. You can see we have hardly any HIV expressing cells. So what we wanted to know was, okay, so you did this is actually an N of like 20 or something. We saw it's about an average of about 50% reduction in HIV expression at this time point in day 6. So just by putting the cells together for 3 days, you can see about half of the cells turn on HIV expression. We also looked at the frequency of infected cells, because one of the questions we always get is, well, if these are CD8 T cells, wouldn't they just be killing the infected cells? Even though they're coming from HIV naive individuals, there's a chance you have some sort of HIV antigen specific CD8 T cell there that could be potentially eliminating the infected cells. And so we use one of our reservoir quantification assays. In this case, it's a integrated HIV PCR. And we show that the infection frequency is the same between these two conditions. So we're not losing the infected cells, we're losing HIV expression. And so one of the questions we frequently get is, do these cells have to touch each other in order for this to happen? Or is this something that they can do from a distance? So we use a transwell. We can take the CD4 T cells in the upper chamber and the CD8 T cells in the lower chamber and ask what happens if this is the same time period of using 3 days of whole culture, but now in transwell. What happens to our HIV expression? So in these donors, you can see this is the monoculture. This is our normal co-cultures. We see the standard decrease in HIV expression. But then now when we have the transwell system, now we actually see an increase in HIV expression. So it seems that at least cell proximity, if not cell contact, is required for this reduction in HIV expression. And again, we confirmed that this is, again, there's no loss of infected cells through our PCR. So these experiments, we did these in collaboration with Brad Jones at Weill Cornell, and he took cells from HIV infected individuals. So now the question was, okay, what happens, or is something like MHC class 1, which is known to be the interactor of HIV specific or any cytotoxic T lymphocyte CD8 T cell, if you take that out, do those CD8 T cells still have some sort of function? And so what he did was he took CD4 T cells from HIV infected individuals. He actually activated them in vitro and then infected one with the, I can't speak, HIV JRCSF. And then they did a CRISPR-Cas knockout of MHC class 1. And the purpose of this was to be able to remove the class 1 from the surface of the infected CD4 T cells and ask, now if we put those cells in co-culture with our autologous activated CD8 T cells, which should have HIV specific CTLs in there, do we still see that reduction in HIV expression? And the answer is yes. So in the negative control, you see that we see a reduction, and they do a slightly different ratio than we did. We do a 1-to-1, they did a 1-to-2. But you can see a reduction in the HIV expression, and these are again coming from HIV infected donor cells. Now, when they knock out the beta-2M, they still see a reduction in HIV expression, meaning that the MHC class 1 expression is completely dispensable for this interaction. So what is the mechanism? We're still working on it, but we have some hints. So one of the things we did was we did some RNA-seq. So we labeled the cells from our co-cultures. The CD4 T cells were sultrates violet, and the CD8 T cells were sultrates red. So after the co-cultures, you can sort them out using the sultrates dyes. And then we did RNA sequencing on the CD4 T cells coming from the monocultures or co-cultures. And you can see this is just looking at the reads of HIV RNA expression. You see in the co-cultures, which are the green, we have a reduction in HIV expression all along with the transcript. So now we actually looked at what the gene signaling was being disrupted through the co-culture. We see the downregulated regulation of a whole lot of metabolic and effector function genes, as well as an increase in apoptosis pathways. I'm sorry, decrease in apoptosis pathways, but increase in cell survival pathways. So we see this is a relationship between the regulation of gene expression as well as compared to the expression of HIV. We see that there is a lot of metabolic pathways that are being downregulated. Glycolysis specifically is one of the ones we're very interested in, as well as a decrease in apoptosis. When we looked further, we see that there's an upregulation of pathways associated with quiescence, memory. This again is a TGF-beta, which is significant for us because that's something that we based our original assay on. We see an increase in TGF-beta responses, and then a decrease in T cell activation. And one of the things that stood out for us was not just the TGF-beta signaling, but also Wnt signaling. And Wnt signaling is increased in the CD4 T cells that came from the CD4 CD8 co-cultures. So we know that not only are things being reduced, but we also see an upregulation of pathways that are really responsible for longevity, stemness, and quiescence of the CD4 T cells. And why this is interesting is because there's a lot of known pathways that have already been described, looking at wnt beta-catenin, TGF-beta, and their role in cell survival in CD4 T cells, and as well as their maintenance in terms of maintaining memory and longevity. So this is actually something that was very intriguing to us. We were kind of suggested that one of the things that CD8 T cells are doing as a regulatory mechanism in terms of promoting quiescence and longevity in CD4 T cells happens at just the wrong time. That basically an HIV-infected CD4 T cell, if it's being subjected to this mechanism, we actually start to see these cells becoming quiescent and shutting down expression just the time you actually want them to keep expressing HIV so they can be recognized. And so how do these CD8 T cell observations potentially impact HIV cure strategies? So it's all great to see all these things in vitro, but you really want to know what is this going to mean? We're going to try to eradicate this reservoir. And so one of the prominent strategies to achieve HIV cure is called shock and kill. And the idea is that if you have this latently infected cell population that can't be seen by the immune system, can you introduce some sort of intervention? In this case, we call it a latency reversing agent, or LRA, to try to wake these cells up, to get them to express some proteins, all in the context of ARC. So they're not, even if they start expressing virus, that virus can go on to do other bad things, but just you want enough virus expression so that these cells can now be signaling the immune system, I'm here, come take care of me. And so the idea is that either through just inducing so much HIV expression in the cells, they die through cell death, or they're actually targeted by the immune system. Eventually, through repeated dosing of the LRAs, you can actually just get rid of all the infected cells. And so the idea of shock and kill is to achieve this broad and efficient latency reversal, but you also want to not be able to have global immune activation because that leads to problems. So it's a very ambitious idea, shock and kill, and to date, it hasn't worked. Basically, there have been 160 different compounds that have been identified as potential latency reversing agents. A number of them have gone into clinical studies, 128 HIV cure-related trials, 99 of them have been interventional, looking at these LRAs, all different classes, looking at all different parts of cell metabolism or gene expression to try to see if you can get this to work. And so far, none of them have done really much of anything in vivo. And so one of the things that really intrigued us about this was if you need in vivo this LRA to work to kind of wake up HIV expression, are CD8 T cells doing something to kind of impact how well that LRA can work? And so a key prediction of our virus silencing hypothesis is that if we take out CD8 T cells, we then see real function of an LRA. And so this was demonstrated in a study that was published a few years ago. This was beautiful work done by a graduate student in Guido Silvestri's group, Julie McBrien, where again, they did using the rhesus macaque SIV model. After infection, they were put on anti-retroviral therapy for 12 months. And then they wanted to use basically a design where they could, one, just look at an LRA. In this case, they're using an IL-15 superagonist. It's just a more stable version of IL-15, which has been shown in vitro that it can actually act as an LRA and reactivate virus expression. So they wanted to put that into the animals by itself. We have the CD8 depletion group, so just taking out the CD8 T cells. Again, all the animals will be maintained on ARC during this time. And then we have a combination. So our hypothesis was that if you're going to remove this virus suppression mechanism of the CD8 T cells, then you should be able to actually see something when you put in the LRA. And so this is the animals treated with LRA alone. You can see this is the, we're looking for plasma viremia that's above our line of detection. And you can see that none of the animals responded to a level of plasma viremia. When we just remove CD8 T cells by themselves, again, the animals are just on ARC. So this is just now seeing spontaneous SIV activation and expression in 11 out of 14 of the rhesus macaques while the animals are still on ARC. Now, interestingly, when we combined this LRA with taking out the CD8 T cells, suddenly we saw robust and sustainable SIV expression plasma viremia, suggesting that one of the things that CD8s were doing was really suppressing the ability of this LRA to be able to act in people. And so, lastly, I'll show you that we could actually recapitulate the same activity in vitro. So we went back to our LARA model system, where I mentioned we can generate a population of latently infected cells. And we can also do the exact same experiment where we can apply a latency reversing agent in the presence or absence of autologous CD8 T cells and ask, do we see the same thing in vitro? And so here's the design of our experiment. We use N803 exactly as they did in the monkey study. IL-15, which is, again, N803 is just a super stable form of that. So we use the parental cytokine, and then as well as just doing TCR activation. And what we showed is this is the reactivation that we see in the CD4 monocultures with the anti-CD3, CD28, the N803 or IL-15. And then as soon as you start adding the CD8 T cells at a 1-to-1 or 1-to-5 ratio, just like we see in the animals, when the CD8 T cells are around, we start to see suppression of the reactivation activity by the N803, even with a strong TCR activating signal like CD3, CD28. We see that the CD8 T cells can really inhibit the reactivation of HIV expression. So I hope it convinced you that we found something really interesting in terms of HIV and CD8 T cell latency activity, but there is still much to learn about this mechanism. So we have the in vitro data from our LAR models. We have the non-human primate models using the rhesus macaque. We have some data from patient samples where we did the MAC class 1 knockout and showed that that's dispensable for the suppression activity. So really what we're very interested in is how this applies to HIV cure strategies. So our data is really supporting a reduced efficacy of the latency reversal activity when the CD8 T cells are present. Well, what are we going to do about that? So our take-home message is that CD8 T cells have two functions in control of our replication in the normal absence of ART. These functions normally work together to either get rid of the virus or shut it off. However, when ART effectively replaces CTLs, we now are left with the suppression activity, and that becomes dominant during ART treatment, and importantly, interferes with interventions to induce virus expression and try to facilitate HIV cure or remission. So projects that we're working on in the lab, there's still a lot of RNA-seq data to pile through. We've done a number of experiments. We're trying to identify the subpopulation of CD8 T cells that are responsible for this effect. We started sorting different populations of CD8 T cells, putting them in cell culture, doing the RNA-seq at the end to look to see what populations of CD8 T cells are modulating those TGF-beta and Wnt signaling pathways. We're looking at epigenetic remodeling of the CD8 T, the CD4 T cells, with the CD8s. And then we also have a cohort of 60 people living, 60 ART suppressed people living with HIV that we're now starting to characterize for latency activity. We have a combination of elite controllers, so people who have a really high potent HIV specific CD8s. We have people that initiated ART during acute infection versus chronic. We have individuals who are not on ART at all, and as well as individuals who've been on short-term ART, so just a few years versus long-term ART. So people have been on ART for like 20 years. And then we're going to be looking at a number of different immune modulators and metabolic modulators in order to better understand how we can see a range of these activities coming from the patient's samples. And so with that, I definitely need to acknowledge all the really hardworking people who are involved in these projects. And I already mentioned Guido Silvestri and Mirko Pagliardini. The work that I showed you and all the in vitro assays was done by Lavinia Franchitti, Simona Mustachio, Hong Wang. The CD8 depletion study, the first one I showed you, where it was depletion at the beginning of the infection, was done by Mara Statsu with Adam Wong. And then the last study that I showed you, which was depletion after a year of ART, was led by Julian McBride. Of course, we had a lot of help from the genomics corps, the veterinary staff who takes care of all the animals. We collaborate a lot with Rafiq and his group here at Emory, who do a lot of the omics analysis with us. We were partnered with Vince Marconi, the Department of Medicine, for recruitment patients. And then we have Shan, who does all of our viral loads. And then I mentioned some of the collaborators we have who do the bioinformatics. Jeff Safry provides the N803, some of the other assays that we used in the quantification of HIV or SIV infected cells, as well as Brad and Talia who did the class 1 knockout work. And so I definitely also would like to acknowledge our funders, the Erase Agent Laboratory and the Center for AIDS Research here at Emory. Thank you.
So in your non-human primate models, what are you using for CD8 depletion for the antibodies?
Yeah, it's a very good question because there's a couple of different versions. So the one that we use is a CD8 alpha depleting antibody.
Okay.
So would get rid of NK cells and anything that has CD8 alpha expressing on it. The antibody is we use one dose and it really rapidly depletes out CD8 T cells. They start to come back about 4 weeks after. But I can tell you that when they reconstitute, they're not the same, right? So initially you've got the mix of naive CD8s with memory CD8s. After depletion, really what reconstitutes is the memory compartment. You no longer have all those naives because you just don't have the virus to make them anyway. So you get CD8 T cell responses. You get CD8 T cell reconstitution is just not the same level. We've also done experiments where we looked at a CD8 beta depleting antibody, which would just specifically target the CD8s and leave the NKs alone. So we get a lot of questions on, well, can NKs do this too? Right. And the problem with the CD8 beta depleting antibody is it's not as effective as the alpha. And so when we look at the tissue distribution of CD8s, we can still find them in lymph nodes, and they're just not nearly as clear to the levels we see with the alpha antibodies.
So can you see tissue resident memory CD8 cells in the beta depletion?
Yes.
But you can't in the alpha.
Correct.
Okay. I mean, and either, because my question was to do with NK cells versus the CD8s, and because you have two groups, right, in that non-human primate model where you have depletion before infection and depletion after 2 weeks of infection. And so my question is like, one of them you're taking both out, and then the other one, you're not taking them out until the end. So I guess, how would you get at the NK cell question?
So we have done a few in vitro experiments, just like isolating NKs and doing that. And I can tell you that NKs do have a suppressive function as well. We haven't quite, we were working with Matt Carson's, who is a big NK cell expert who left. So now we're looking for more NK cell experts. So who can really help us with that, because we did see some suppression activity just from our in vitro cultures.
Yeah, I guess my biological, the reasoning behind that though is because like the NK cells are going to be activated first. They're going to show up in the first like 2 to 3 days, and then the cytotoxic cells aren't going to show up until the, like day 7 or 8. So there could be some suppression there, like by the NK cells in the first few days of infection in the group, if that isn't depleted until 2 weeks after.
Yep.
Okay, great work, thanks.
So we're seeing one of the things that we're very interested in, and why we think this has some in vivo relevance is there are a number of different cancer studies that have identified CD8 regulatory populations that really impact the effectiveness of immune responses like CTLs or tumor infiltrating lymphocytes. It can block their activity and really kind of suppresses a lot of effector function. And so these cells have been shown to be useful targets for cancer therapies. You basically try to suppress their suppression activity and basically allow for the immune system to function a lot better. So we're kind of taking a cue from cancer research, which is something HIV does all the time, and looking at some of the things that they have been targeting, some of the pathways and compounds that they've been using in those sorts of studies, so we can look at them in our in vitro systems and then apply them to the HP.
Anyone else?
So I was just wondering, what is the role of CD8 T cell expression in the effect that you are like the regulatory effect of CD8 T cells that you are seeing in the ART subjects, ART treated individuals. So is CD8 T cell expression playing a role in that regulatory effect?
That's a very good question. We haven't gotten all the way there yet. So in our vitro model, we have characterized all your negative regulators, PD-1, I can tell you that we do see a high increase in negative regulator expression on the CD8s in the in vitro model system. But in that context, where they're not exhausted, they're just activated. So our in vitro, that's our real, limitation of the in vitro models. We can't really model exhaustion like you can with someone who's been on ART for a year or the NHP model. So we're really have to pull the cells out of the freezer from the NHP studies to really look at that more. Some of the things that we're obviously interested in are tox. And PD-1, neurocardini is doing a lot of that work. And so we're characterizing the expression levels of those markers on the suppressive versus cytolytic CD8 T cells, and to try to better understand, are some of those actually markers of not necessarily exhaustion, but suppression. And so that's what we're looking at.
Oh, go ahead.
Sorry, this is a more general question, but I don't know how prevalent the usage of ART is worldwide. But do you know if we're just like selecting for HIV strains that are more likely to be dominant all the time?
That's an interesting question. My guess is no. And the reason I say that is because when we see latency, the mechanism of latency is, and I'm just gonna give you my pet theory for a second here. The mechanism of latency, people have this sort of thought in their minds, like, what's going to happen is you're going to have a primary infection, CD4 T cells are going to be activated all over the place. The virus loves activated CD4 T cells. It goes in. Some of those cells just turn into virus machines, and some of them, eventually shut down virus expression and then become the latent reservoir. I personally don't believe that mechanism is very plausible. And so, especially given a lot of the priming data we see, you can see latently infected memory CD4 T cells in lymph node 3 days post-infection in a non-human primate. There's no way that cell went through activation, productive infection, and then, quiescence in 3 days. We have a lot of memory cells that are being infected and then establishing a latent infection right out of the gate, which is why when we're doing that primary acute infection, we have that mix. So I think that there's both going on at the same time. But I have yet to see any evidence that that we're selecting for viruses that basically shut down faster than others. What I think we're going to see, and this is basically hard to do unless you actually ask people to do this, but there's been some ideas that treatment of ART could potentially, in some people, basically allow a reset of the immune system. So that if you look at, and this is why we're interested in T cell metabolism, is because you initiate ART and then you let them go for a while. Most of the people, you're going to see that exhaustion, right? You're going to see this chronic exposure to viral antigens, and you just basically have this sort of immune fatigue. But in some individuals, they're not frequent, but there's some of them that actually do immune system reset during that ART time. And if you take them off ART, suddenly they look like elite controllers. Like they don't viral rebound. They have beautiful control of plasma viremia. So those are called post-treatment controllers. We have yet another group. And studying the immune system of these individuals is really important, because if you look at those individuals right before they go on ART, they're indistinguishable. Like everybody looks the same. Your CD8 T cells, they're not better than the other. You don't have more infection or less. Everybody looks the same phenotypically, but somehow when they start ART, some people do a reset. And that gives me a lot of hope that if we can identify what is driving that reset, that we could actually make everyone. And so that's one of the things we're doing with one of our collaborators, Azir Sirion, who's very big into T cell metabolism. And he's identified a number of different pathways that are involved in that reset. And so we're trying to replicate that in vitro.
So more of a clarifying question. So when you're depleting these T cells and then seeing sort of viral rebound in animals, but they're on ART, are you seeing they're on ART? So you're not seeing new replication cycles. You're seeing replication of RNA basically in cells of the idea.
Mm-hmm.
So there's is it? I guess I'm just wondering, is it tolerated well by the animals? Is there clinical? Do clinical scores indicate that they're sick?
We haven't lost any animals during that time. So basically the plasma viremia, while they're on ART, because of the fact it's not spreading infection, it's just like isolated events of cells starting to express SIV. They actually do okay. The plasma viremia is not the level if we just took them off ART completely and then the plasma viremia shot through the roof. So it's well tolerated by them at that level. And one of the reasons why we're very interested in that is you want to find the level of plasma viremia that can be therapeutically interesting, because then you want to be able to like, this is the level that everybody can do okay at. And that gives us an opportunity to come in and like sort all those virus expressing cells.
So, in thinking about clinical applications of this, specifically the latency reversal agents and CD8 depletion simultaneously in humans, like especially with like the IL-15 superagonist, that's going to activate like all CD4 cells. And so what would you think would happen in like secondary infections or in people who have infections like with TB or flu or something like that? And side question was, do also lung tissue resident memory CD8 T cells deplete as well?
And this is one of the big reasons why LRAs they're a great concept in a dish and not so great in others. And so that really ambitious goal of how do we find something that stimulates HIV expression but doesn't like blow the rest of the immune system up? It's been really challenging to do, and thus far none of them have worked very well. I think that there's a combination of both the CD8 suppression mechanism as well as putting in the drug in a dish on some cells is not the same thing as administering it in a deep. So I think that IL-15, no way it's going to happen. We're interested in there's a few drugs, again, we're borrowing a lot from cancer in terms of things that have kind of worked on that side. But for sure, that's one of the things we really need to look at is because you're applying the drug systemically, but you can't. And one of the other things we're very interested in is potential targeting, like coupling the compound with some sort of like a mimetic or something that can actually target a particular cell type that we can go over there. And we thought about that. That's one of the reasons why we really want to identify the CD8 T cells, is potentially we could do like a suppressor or activator of those particular subpopulations. So.